Hello everyone, and welcome to the second iteration of the DinoSphere online seminars. So these seminars are part of an effort to bring dinoflagellate enthusiasts of various fields together. You can read more about it on our website that you should find in the invitation email, but we can also share it here. The important thing here is these seminars are really focused on early career researchers. So we're trying to invite PhD students and or postdocs like today, Edmée and Nicolas, who will speak to us. And we also really try to emphasize that people talk about what there maybe hasn't worked or was really difficult in their research, and which might also encourage then other people listening to, ask questions that can help them in their own projects. So we really want to encourage discussions about everything. So we're, fine if you also present things that are not didn't work out so well, or that you had struggles with, because we all are struggling with dinoflagellates sometimes. So anyway, to make this short, the important thing is we are recording these seminars from now on, because it's just impossible to find the time that everyone in the world can join. And the seminars will be made available for 2 weeks on YouTube after the talk. And if you should miss that and really want to hear it again, then you can contact us and we will ask the speakers if they're okay with sharing the recording. That's mostly from me now. I will hand over now to Liz, who will introduce the speakers, and I will come back to you at the end. Thank you.
Thank you, Lily. Yes, so I just want to introduce Edmée Royen, who is, I believe, just finished her PhD at University of Liège, and she is going to talk to us about the work she has done on Symbiodinium. So, Edmée, if you would like to go ahead.
Thank you for the introduction and for the opportunity to talk today here. So yeah, as you said, I just finished my PhD in the lab of genetics and physiology of microalgae under the supervision of Pierre Cardel. And yeah, I'm here to talk to you about my favorite dinoflagellates, which are Symbiodiniaceae. Um, you all have already heard about Symbiodiniaceae because they form a mutualistic partnership with corals, and together they are at the basis of the coral reefs, which is one of the most biodiverse ecosystems on Earth. Symbiodiniaceae are particularly important because they conduct photosynthesis and they give a large proportion of the products of photosynthesis to their animal host, and this accounts for a large proportion of the primary production of the reefs. But Symbiodiniaceae can also establish symbiosis with other organisms, including jellyfish such as Cassiopeia, and this is how I met Symbiodinium during my PhD thesis. In brief, we took our jellyfish, we subjected them to a moderate thermal stress, and we measured several jellyfish metrics, extensively photophysiology, and we conducted bottom-up proteomics on both symbiotic partners. But it's not the topic of today, so I'm not gonna go over it further. But if you have any question about this Cassiopeia story, don't hesitate to ask. Symbiodinium was first described by Hugo Freudenthal in 1962 from Cassiopeia zooxanthellae, by the way. But given its ecological importance, it has been widely studied since then. There were several clades which were described, and now they have been redefined into genera of the family Symbiodiniaceae. In common, Symbiodiniaceae all have a type E eye spot. They have a mastigote life form, but also a coccoid life form. The mastigote has a series of 7 longitudinal amphiesmal plates, and they have a single perineurid with a starch sheath and no thylakoid extension in the inner matrix. In general, I think Symbiodiniaceae are rather small for dinoflagellates, from 5 to 10 micrometers, and have a rather small genome, nuclear genome, given that they are symbiotic. I mean, it's a trend. But I was not so interested in morphology. I was interested in photosynthesis. So as many of you know more than I do, in dinoflagellates, you can find a wide variety of plastids. In the case of Symbiodiniaceae, they have the canonical peridinin plastid, which is already very specific compared to some other plastids. It's surrounded by 3 membranes. Thylakoids are stacked by 3s. The main photosynthetic pigments are chlorophyll A, C2, and of course peridinin, which is unique to the dinoflagellates. And their plastic genome is highly reduced into 10 to 20 genes, which are organized on circular DNA molecules, which are called minicircles. Symbiodiniaceae are photosynthetic, but they are also heterotrophic. Here, there is this nice picture of ephrenium that is puncturing an ochrophyte. But as you know, approximately half of the described species of dinoflagellates are not capable of photosynthesis. They are solely heterotrophic, and this is because they have lost the peridinin plastid throughout their history, and this is also one of the reasons why there are so many different plastids in dinoflagellates. This loss of photosynthesis is what I have been studying during my thesis, part of my thesis, using the model Symbiodiniaceae. This project has been initiated actually by Adrian Barbrook, Ellen Nisbet, and Christopher Ho from the universities of Cambridge and Nottingham. They have been isolating strains of Symbiodinium microadriaticum by growing them on supplemented medium, supplemented with glucose and amino acids. The goal was that these conditions would give alternative to photosynthesis for these mixotrophic organisms, and that it would make photosynthesis optional. In these conditions, colonies with different colors and different sizes have spontaneously emerged, and we have isolated these colonies and called them GA for glucose amino acids. Given that they were likely impacted in their photosynthesis, we investigated their plastid genome. So what I didn't tell earlier is that the genes encoded still in the plastid genomes are all related to the photosynthetic electron transport chain. So is either subunits of PS2, PS1, cytochrome B6F, or ATP synthase. By performing outward facing PCR on the mini circles, we have observed that some strains were missing mini circles with either PSBE or PSBI, and some strains were defective in the mini circle with PSBA. So these proteins have already been characterized in other photosynthetic organisms, so we already know what they are useful for. So PSBA is working in the reaction center of PS2, and PSBE and PSBI are involved in the assembly and repair of PS2. So it's already interesting to see that all the mutations are related to PS2. Therefore, we looked at the activity of PS2 through chlorophyll A variable fluorescence, first measuring the maximum quantum yield of PS2 or FVFM. For the wild type, we get something around 0.55 or 0.6, which is expected in Symbiodiniaceae in this context. And for all the other strains, we get a value that is lower. So this is already interesting to note that all the spontaneous mutants are impaired in their PS2 activity. We wanted to focus on 2 strains, so we chose GA1, which is missing PSBI, and GA2, which is missing PSBE. So first, to characterize them, we looked at their growth on different medium and at different light densities. When we look first at the wild type in F2, so the minimal medium, we see that it can grow with light and it grows more with increasing light, which is expected. But on medium supplemented with glucose and amino acids, there are several things to say. First, we can grow these wild types in the dark in complete heterotrophy. And then we see that it grows better under higher light. There is an improvement of the growth. However, this improvement is limited in high light intensities, which involves that there is a relationship an interaction between photosynthesis and the use of glucose and amino acids. GA1 can grow on minimal medium, but less than the wild type, and it can grow similarly as the wild type on supplemented medium. GA2 cannot grow at all on minimal medium, and it grows less than the 2 other strains on supplemented medium. What's nice to see is that it grows better under low light than under no light, which means that even though it cannot grow in fully autotrophic condition, it still can take advantage of light somehow. So we wanted to further characterize the activity of photosynthesis in this strain. So we measured the relative electron transport rate through PS2, which is a measure of the activity of PS2. For GA1, when we look at the activity of PS2 as a function of light intensity, we see globally the same kind of curve as in the wild type, but with a lower maximum value, which indicates lower PS2 activity. In GA2, no matter the light intensity, there is no activity of PS2, which confirms our measurement of AVFM. However, when we look at the maximum fluorescence of each strain, we see that it's not zero for the mutant strains, even in GA2, which likely means that there are some kind of PS2 assembled structure, partially assembled, which are partially functional or not functional at all in the case of GA2, yes. We also investigated the activity of PS1 by measuring the variation of absorbance at 705 nanometers. First, when we look at the wild type, we have a high value under standard condition, but when we add PS2 inhibitors, this value is clearly decreased. This is expected in the case of a linear electron flow. So as you know, the electrons that are taken from rotor by PS2 are transported through the plastonic-Kinnon pool, cytochrome B6F, PS1, and they are eventually used to reduce NADP plus into NADPH. And together with this electron transport, there is a transport of protons from one side, to the lumen of the thylakoids, which can be used to produce ATP. So it makes sense that if we use PS2 inhibitors, the activity of PS1 decreases. But the activity of PS1 is not zero either. And this PS2 independent PS1 activity is an indication of circular electron flow around PS1, where reduced molecules at the level of PS1 are used to reduce the plastonic-Kinnon pool, and then it goes back to PS1, which is useful to transport protons to the lumen of the thylakoids, and then potentially be used to produce ATP. And this process is actually happening under normal conditions. to make the balance between the production of NADPH and ATP. In GA1, we see that there is an activity of PS1 that is lower than the one of the wild type, which is expected because the activity of PS2 is lower. And again, when we add inhibitors, the activity of PS1 decreases when PS2 inhibitors in a similar way as the wild type. So probably there are both circular and linear electron flows, but in different proportion than in the wild type. In GA2, there is a low activity of photosystem 1, but it's not zero. And it's exactly, the same in the presence or absence of PS2 inhibitors. And this likely indicates that there is a cyclic electron flow and only cyclic electron flow occurring in this GA2 strain. So in a word, in GA1 that is missing PSBI, there is a reduced activity of photosystem 2, which has a negative impact on the growth on minimal medium, but not on the growth on supplemented medium. GA2, which is missing PSBE, does not have any active PS2, but has PS1 that allow cyclic electron flow. This is not enough to grow on minimal medium, but that can have a positive impact on the growth on supplemented medium in low light. So I was asked to, yes, all in all, our main conclusion is that our spontaneous mutant all lost photosystem 2, and then therefore the loss of photosystem 2 could be a first step towards the loss of photosynthesis in Symbiodiniaceae, but also maybe in all peridinian plastid dinoflagellates. And the retention of PS1 could be an intermediate step that could allow that allows here cyclic electron flow, and that could be useful to produce ATP. So I'm gonna make it short. I was asked to talk about the challenges about working with dinoflagellates, and I have to say that in my case it was far easier to work with Symbiodinium than with jellyfish. But yeah, I would like to bring points that are interesting in Symbiodiniaceae that might make it a model dinoflagellate. First, several species are easy to grow in the lab on different media. Axenic strains have been isolated. Genomes and transcriptomes are available for several species. Molecular tools are also coming up right now, such as nuclear transformation, and there are useful protocols such as protocols to make protoplast or cryopreservation. So there are challenges, I have to say. There are still many uncultured species, so we don't have a good idea of the diversity of Symbiodiniaceae. And from my point of view of a person that has been working on photosynthesis, it doesn't work exactly as in the green lineage. So many things still need like a definition, proper definition, and especially in the photoprotection, things don't work exactly the same. So further investigation is required. So I'd like to thank you for your attention and thank you for allowing me to speak today. I'll take any question.
Thank you, Edmée. That was a great talk. So yes, if we have any questions, I think we have time for that. Plenty of time.
I'm just showing this.
Can I ask something? And I really like that you can grow them on plates. Actually, I've never really thought about that. Did you try something similar with other dinoflagellates? I mean, not just growing them on plates, but also trying to force them to give up photosynthesis?
I never tried.
Yeah.
I don't know if Chris or Lisbeth and Adrian Barbrook ever did it, because I know they work a lot with Amphidinium. I think it would be really nice if we could do it, but I didn't.
Yeah. Okay. Yeah. No, it's super cool. I kind of like that you have little dinoflagellate colonies and nice.
Yeah,
Good
it's super
for
useful
you.
to isolate stuff, yeah.
Yeah.
It looks like we have a couple questions. First from Noriko.
I think Richard should be first. I'm kind of organizer, so.
Oh,
Okay.
okay. Actually, I just had an answer to the previous question. For Amphidinium, you can plate it for about 3 hours, but it doesn't like staying on plates. And I think it's something that's quite exceptional about Symbiodiniaceae. And also with other coral symbiotic algae, such as Chromeria also, it is plateable, but for the majority of dinoflagellates, they don't like that. So this is a great system, therefore, for developing gene transformation and functional genomics techniques. And I'm sorry, I didn't have a question, but I just wanted to say, Manan, that your work is really, really, really cool, and thank you for sharing it. Sorry, Esme, really, really cool, and thank you for sharing it.
Thank you for this. I didn't know.
Interesting. Okay.
Okay.
Noriko?
May I give you a question? It was very great talk. I really like it. Thank you so much for talking about this photosynthesis topic. Actually, I think you already mentioned during presentation the mutated genome, the mutant could be back to the photosynthetic species under the high light condition. Is it right? Or they keep the heterotrophic or mixotrophic condition, even you bring back them to the normal light condition.
So
Do you understand my question?
I'm not sure what it means.
I wonder
Mm-hmm.
if your mutant chloroplast and photosynthesis ability can be back to the under the normal light intensity condition.
Like if I put these mutants that are defective, like if I put them back to normal light intensity, could
Exactly, yes.
they...
Yeah.
No, usually no. Like when I grow them, here, for example, at 50 microeinstein per meter square per second. I mean, these are not a good example, but yeah, here in F2 they will grow less well than the wild type.
Okay.
So
Okay,
they
it's
won't
interesting. So
reverse.
it means you exactly make this mutant artificially, and do you think this heterotrophic, how to say, losing to the chloroplast or photosynthesis ability almost never happened in nature, natural condition? What do you think?
Actually, I think that this happens often,
Often.
actually, because well, I don't have numbers, but we obtained them in a rather short time, and we just isolated them on supplemented medium. So I think the mutations were already there. And by putting them in a medium where photosynthesis is less useful, the mutant could grow, and especially because they had this food to compensate. But I think that usually when you have them in flask, the mutants would die out and not like gain over because they are less successful. I think this must happen uh, like quite often, but
Yeah.
it's
Makes
hard
sense. Yeah.
to
It's
measure.
very interesting. If
Yeah.
may I ask one more question? I think this time you focus on the photosystem, but did you also measure the Calvin cycle activity? Or
No.
you plan to, maybe?
It's something that would have been very nice, especially in this GA2 mutant. Because yes, I don't know if I can find this photosynthesis thing, but yeah, it would have been nice. Probably in GA1, I think it does work, but we didn't measure it simply because it can grow with on minimal medium and with light. But in GA2, it would be really interesting because there is likely only ATP that is produced and no NADPH. So if the Calvin cycle works, it means that there is like reducing power that comes from somewhere.
Wow,
And so yeah, that would be interesting. But we didn't do it. We didn't have time during my PhD.
Okay, yeah, thank you. Yeah, if cyclic, only the linear, almost a linear never works, probably NADPH never produced. So yeah, makes sense. Yeah. Thank you so much for interesting talk.
Well, thank you for your questions.
Lily, do you have another question?
Yeah, sorry. It's really cool. Do you know how many of those colonies were isolated and screened? Because it's kind of curious that there is like but 3 different proteins, photosynthetic proteins affected kind of specifically.
So I didn't do the isolation myself, so I'm not actually sure of the numbers. But I know in the first shot of this experiment, there were these 7, but they are not shown all here. But there are 7 mutants that had this anomalous phenotype. Yes. So not all genes were impacted. I think there are reasons behind that. I don't. Yeah, let's start by the easiest. I think PSBE and PSBI are found in lower copies than the other mini circles, and therefore, when there is this, how would you say, like when the chloroplast is replicated, I mean the genome is replicated, there might be bad repartition, distribution, of the mini circles, and I think this is how PSBE and PSBI are lost.
Okay, cool.
And for PSBA, I'm not sure actually. I don't have far more than this so far.
Okay, yeah. No, thank you. Thanks a lot. Oh, there's another question in the chat.
Yeah. Okay. So this question is, I have a question on the colony appearances under various conditions, slide 9. With wild type under 100 micro-E light, for example, do we know adding amino acids and GLC reduces cell density, or maybe the cells simply make fewer pigments?
I measured the pigments per cell. I think it's a little higher than the wild type in GA1, and it's a little bit lower than the wild type in GA2, but nothing is crazy. So it's not only a question of pigments per cell. There is really a difference in the amount of cells that grow. I'm not sure I got the whole question. I think I answered just part of it.
Yes.
Yeah, so I was wondering, so so we can see the brown is more brown than the less brown ones, but I'm just wondering whether it's possible that the cells are simply making more pigments, like chlorophyll AC or pyridinine or carotenoids, that makes the cell colony look denser. So I was wondering about that.
Yeah, no, basically the amount of pigments is similar between the strain. It's lower in GA2 than in the wild type. But I also counted the cells, so I know that here there is also less cells than here. And yeah, the cell size also is the same, so I think the impact is not so much on the amount of pigments.
Okay, that's it. Thank you. And then for the wild type, why do you think adding the hydrotrophic media reduces the cell number at 100 microangstrom?
But I didn't investigate it so much, so it's hard for me to tell. So in the first part of the answer is exactly what you said before. Probably there is a decrease in the pigment number, because I said that I counted the amount of pigments, but just in this condition at 5 microangstrom per meter square per second. So in this case, that I think that the pigments could decrease. But I think also maybe if photosynthesis is not as controlled as it is, because there is this other, carbon and nitrogen that are given by another way. I think that there might be a production of rust that is less well controlled, so maybe it could impact the growth.
Yeah, that makes sense. If you have less pigments, yeah, maybe rust just kills the cell more easily.
I didn't check, I have to say.
Okay. Okay. Thank you for the talk.
Thank you.
It looks like, let's see, maybe time for this one last question from Richard. It says, I wonder if you have looked into the presence of PSBT, which may be a new and less essential plastid-encoded dinoflagellate protein in your mutant lines. And I will say that he also responded a little bit to what you guys were talking about in the chat, if anybody's interested.
Okay, I will go check. But no, we didn't check PSBG. I did not actually. So I don't know.
Okay. All right. So I think it is time to move on. Thank you so much, Edmée. And so yes, our next speaker will be Nicolas dos Santos Pacheco, who is working with Perkinsus at the University of Cambridge. And so, Nicolas, if you would like to, let's see, we have to switch screens, I think, first. Edmée, if you wouldn't mind unsharing. Perfect. Thanks.
All right. Can you see my screen now? All right. So yeah, today I'm going to talk about Perkinsus, which is actually not a core dinoflagellate. And actually, I'm not used to hear a lot about dinoflagellate talks because I did my PhD on apicomplex and parasite, which are actually related, but not that close to dinoflagellates. So Perkinsus is a marine parasite that infects certain types of mollusks that are called bivalves, like clams, cockles, oysters, and mussels. And those bivalves are very important for two main reasons. First, they are the source of food for a lot of people around the planet. So they are of great economic importance. And the second is that those bivalves are filter feeders. So they are filtering the water to get their nutrients. So they are getting the water cleaner, and doing so, they are sustaining the whole coastal ecosystem to thrive. And so today I'm going to talk about Perkinsus chesapeake, which is a species which is found on the east coast of the US and also on the all around the Atlantic. And that is infecting the clam and the cockle mainly, but is also found in the Pacific oyster. So Perkinsus is part of the Perkinsozoa phylum, which is a sister group to the dinoflagellate, and which is related to the apicomplexan parasites. And those apicomplexan parasites, you might have heard of them. There is Toxoplasma for toxoplasmosis. There is also the malaria parasite, for example. So the Perkinsozoa phylum is composed only of parasitic species, and it's actually four groups of parasites. So there is a Perkinsus that infect the mollusks, but there is also the group of Paravillicifera and Marantos that are infecting other dinoflagellates. There is X cells that are infecting fishes. And then there is also what we call the SPI agents that are infecting tadpoles, frogs. And so in addition to the economic and ecological importance of studying Perkinsus, there is also this evolutionary importance because they are related to apicomplexan parasites that are medically important. And so by studying the Perkinsus, we can understand how parasitism arised in those two independent groups from a common ancestor that was probably not a parasite. And so one of the hallmark of parasitism in apicomplexan parasite is the presence at the apical pole of the cell of this apical complex. And this apical complex is composed of cytoskeletal component with a conoid at the center, which is made of tubulin, and also some secretory organelles that are called micronemenrhopteres and that are important for the invasion process of the parasite. So this is very, very important for the life cycle and for the pathogenesis of those parasites. And so this is a complex that I studied during my PhD. And so I was quite amazed when I collaborated with Ross to write a review to know that there is actually apical complex outside of apicomplex and parasites. And so here, for example, in the zoospore stage, which is the flagellated stage of Perkinsus, there is a similar apical complex found at the apical pole of the cell. In this case, the conode is also made of tubulin, but the conode is open on one of the sides. And there is also some secret organelles that are also called micronemenhopteres, even though we actually don't know what's inside those organelles and what is the function of those organelles in Perkinsus. And so this is the main goal of the project, is to understand the composition and function of the apical complex in Perkinsus. And by doing so, we can get some insight on the key component that enables the transition toward parasitism in both Perkinsozoa and apicomplex and parasites. And so if you want to study the apical complex of Perkinsus, we first need to get access to the zoospore stage. And this is not an easy task. There is not a lot of cell biology done on Perkinsus. And the vast majority of it is done on these vegetative stages. So basically trophozoite that will go undergo schizogony to make schizont and then other trophozoite, and we can keep those cells in culture very easily. If we want to get zoospore, we first need to get the trophozoite to transform into the hypnospore stage. This usually happens only in the host, and it's only very recently that there is a team in Japan that developed a media that allow this trophozoite to hypnospore transition in vitro without the need of any host. So I optimized this protocol for the species that I'm working on, Perkinsus chesapeake, and it worked very well. And then when you get this hypnospore in cultures, you can put it back in the regular culture media that usually support the vegetative cycle. And this will trigger the zoosporulation process. And so this is what you can see on this video here. So you have a single hypnospore. The cell is going to contract, detach from the cell wall, and then it will undergo several binary divisions until it reaches the zoospore stage. The process roughly takes 8 to 9 hours, and each division, they are not synchronous, but they take roughly 30 to 40 minutes. And you will see at the end of the video, you can tell that the zoospore are fully formed because they start just moving around inside the sporangia. And at this stage, I was just waiting for this plug here at the bottom of the sporangia to break open. And so they can be released inside the media. And this is what you can see in this video, just zoospore swimming out of the sporangium. And so this process from when we get hypnospore and we put them in the regular media to trigger zoosporulation takes quite a lot of time. So actually, it's only at day 3 that you can see some first sign of division. And it's only at day 5 that you can see some zoospore that are actually egressed out of the sporangium. And in the case of the culture that we have in the lab, we reach a plateau at around 60% of the sporangium responding to this change in media, and that triggers zoosporulation. And so this was when it worked, because I also had some issues with cultures, even if it's not dinoflagellates. And so I could see that roughly after a year of keeping the parasite in cultures, I could see this efficiency in zoosporulation clearly decreasing over time. And it was for like a 1 year cultures, it was decreased only like around 10% of the cell were responding. And so I went a little bit into a panic mode at this stage because I really need to get those zoospore and I need a lot for the experiment I'm planning. So I was wondering what was happening. My best guess was that the cultures were adapting to the in vitro cultivation method. And so they might have accumulated mutation in some of the genes that are important for this zoosporulation process. And so because there is still some zoospore formed, even in those very old cultures, I wondered if I could just purify the zoospore, wait those zoospore to transition back to the trophozoite stage. And this has been shown actually very recently that zoospore kept in culture for a very long time can transition back to the trophozoite stage and then just start new cultures from there and see if these new cultures, so what I call the zoospore-derived cultures, are more efficient to do zoosporulation. And thankfully, this is exactly what happened. So culture that are zoospore derived are again very efficient in making new zoospores. So this was quite a relief. And so actually, before starting to talk about the data on the apical complex, we took advantage of having now all those different life stages in culture. And we can also purify them because they are very different in size to do some proteomic and transcriptomic analysis. So we purified all those stages and did some quantitative proteomic and some RNA-seq, bulk RNA-seq in this case. And so with those data, we did some differential expression to know which transcript and which protein are expressed in all those different stages of the parasite life cycle. We also did some domain enrichment to know which protein domain are specific for one of those stages. So for example, for the zoospore, as expected, we found that all the flagellum-related domains are specific to the zoospore stage. And there was also two other domains, so astacin, that are peptidases, and those pathogenesis-related PR1-like domain that are important apparently in host pathogen interaction in other organisms. Those two domains seem to be enriched also in the zoospore stage. So they might be at the interface between the parasite and the host. And finally, another outcome of doing those RNA-seq is that we're able to update the genome. So we now have 6,000 new genes in the genome of Perkinsus chesapeake. And thanks to the support of this new RNA-seq data, we're able to correct the model of 16,000 genes in the genome. I also did some expansion microscopy to look at the zoospore in more detail and see if I could see the apical complex structures. So this is a zoospore stained with an acetylated tubulin antibody. So we can see the subpellicular microtubule of the parasite and also the two flagellum. And if you look just at the section of this image, you can clearly see the conoid here at the apical pole of the cell. We also stained the zoospore with NHS ester, which is a stain that is going to illuminate the different structures depending on their on the density of protein and the accessibility of those compartments. And I hope that you can see here that there is a lot of stringy and elongated organelles that are going toward the apical pole of the parasite, and you can see them on this maximum projection, those very elongated organelles that are actually the rope trees and the micronyms that have been described by him decades ago. And so in comparison to the apicomplexan, rope trees and micronyms, it seems that those ones are directly docked to the side of the conoid, while in apicomplexan parasite like Toxoplasma, the rope trees and the micronyms are going through the conoid to be secreted outside of the cell. And so to increase even more our resolution, we did some cryo-electron tomography to look at this apical complex. And so here you just have a tomogram going back and forth inside the apical complex. You can see the conoid wall here. And you can see that actually there is several types of potentially secretory organelles, some that are electron dense, some are not. Some seems to have a neck and bulb shape, which is very reminiscent of the rope tree bulb and rope tree neck in apicomplexan parasites. And more importantly, we can clearly see on those tomograms that indeed those organelles seems to be directly attached to the conoid here on this side of the tomogram. One of the other characteristics of the conoid in apicomplexan parasite is that it's not made of canonical microtubules that are made of 13 protofilament. When you're looking at the cross-section, it's actually made of what we call conoid fibers with only 9 protofilaments, and this is a hallmark of the apicomplexan parasites. And so I wanted to know if it was a case also in Perkinsus, if the conoid was also made of conoid fiber or is there canonical microtubules. And clearly by doing subtomogram averaging, we could see that actually the conoid in Perkinsus is made of canonical microtubule, so which is also a striking difference with the conoid of apicomplexan parasites. And so finally, to get insight into the composition of the apical complex, we performed spatial proteomics using hyperlopids. So in this case, we harvested a large amount of zoospore that we basically break open using nitrogen cavitation to release all the subcellular compartment. Then we lay out those subcellular compartments on a density gradient, we spin them down, and this is going to have for effect to separate all the subcellular compartments according to their density. And then we can take several fractions of this density gradient for analysis. Then all those different fractions, the protein are digested, and they are all labeled with a unique TMT label, which is an isotope tag. Then all those samples can be pulled together and sent for mass spectrometry. And then the mass spectrometry is going to have like three effects. First, we're going to detect which protein are in the fraction. We're also going to identify the fraction thanks to the TMT label. And finally, we will be able to quantify each protein across all those different fractions to form those different graphs of yeah. And then we can also reconstruct this 2D map. And basically, this is where the logic of the LOPIT applies. All the proteins that are in the same compartment will usually follow the same profile of quantities across all those fractions. And when you look at the 2D map, proteins of the same compartment are going to form cluster in the map. And so this is the resulting map of our experiment. So for the zoospore of Perkinsus sapieci. So all the dots on this map are proteins, and all the colored ones are proteins for which we can assume the localization because those proteins are conserved in other organisms. And as you can see, the protein of compartments are usually clustering together, meaning that the experiment worked well. And so now the question is, in this map, where is the apical complex? So what do we know about the apical complex? We know that actually the composition is unknown. So we're expecting the apical complex to form clusters without any of those colored markers. And this is what you can see here at the bottom of the map. There is several clusters that don't have any markers, so for which we don't know the origin. We know from the apicomplex and parasite that because those organelles are potentially secretory organelles, they should be enriched in protein that have signal peptides. And indeed, when you look at those clusters at the bottom of the map, it seems that they are indeed enriched in protein with signal peptides. So those clusters potentially represent secretory organelles. We also know that this apical complex is very specific for this zoospore stage. So when we look at our RNA-seq data, we can see that indeed those clusters here are enriched in protein that are zoospore specific. They are in red in the map. And as a confirmation, we can also see that this part of the map here is also appearing to be enriched in zoospore specific proteins. And this makes sense because this is a cluster where flagellar proteins are. And finally, we could look at those domains that we found to be specific for the zoospore, so the PR1-like domain and the astacine domains. And it seems that the proteins that have those domains are indeed resident to those clusters at the bottom of the map. And we're also able to look at orthologs of known proteins in apicomplexon to be at the apical complex. So in Toxoplasma, for example, arrow in green here is a protein at the surface of the rhoptries. And DCX is a protein which is at the surface of the conoid. And the orthologs of those two proteins in Perkinsus seems to also belong to those clusters here. So now with all those data, we really think that all those clusters here represent the apical complex of Perkinsus, and this represents almost 900 proteins. And so, yeah, in conclusion, we now have a cell culture method to get all those different cell stages of Perkinsus in cultures without any host. We now have a specific stage-specific proteome and transcriptome, and we'll have a new and updated genome. We also did some expansion microscopy and cryo-electron tomography to have higher resolution on the apical complex. And we have the spatial proteomic using hyperlopit that is telling us that help us identify the apical complex component. And the next step of this project is going to be to validate this lopit map. And so I try to do some genetic manipulation of the parasite, and actually it's not great. Actually, it's not optimized for manipulation of the zoospore stage. So now I'm going to transition and start to do some specific antibody to certain proteins of those organelles and see if to those clusters and see if they are indeed in these secretory organelles that I can see by expansion microscopy. And with this, I would like to thank people in the lab, Scott that helped me with the hyperlopit, Victor with the bioinformatic, and also Josie Ferreira and Alice, her PhD student back in London that did the cryo-electron tomography. And thank you for your attention.
Thank you very much, Nicholas. That was great. Okay, so if people have questions, we have 10 minutes. So ask away. Lily.
Me again. And yeah, this is super cool. Also, how did you separate the different life cycle stages? You said they have different sizes, but how exactly did you do that?
Yes.
I'm sure it was partially challenging.
Well, actually, I'm just using sieves of different sizes. So for example, the zoospore are the smallest one. They are roughly 5 micrometer. So if I just use a 5 micrometer sieve, only the zoospore are going to go through and
Really?
not the other stages.
Okay.
Yeah. Then the trophozoite are around like 10 to 15 micrometer, so they're a bit larger. And then the hypnospore and sporangia are roughly 40 micrometer. So they are way bigger. So I can actually separate all of them. Yeah.
Okay, that's very useful. It's a very nice
Yeah.
system.
Yeah.
Yeah, I was impressed that you could separate them so nicely. And you just mentioned at the end now that the genetic manipulation is challenging in the zoospores.
Yeah, so actually there
Is
is
there any
some tools
background?
already for
Mm-hmm.
Perkinsus, but they are all have been developed for the trophozoite stage.
Okay. Mm-hmm.
And so actually there is some issues with the promoter that I can use because if I want to express a fluorescent protein in the zoospore stage, I need to have a promoter that is active in the zoospore,
Okay.
but also in the other stages because the transfection is done at the trophozoite stage. So I need the trophozoite to express the fluorescent protein, and then I would trigger the zoosporation process until the zoospore and see where the protein is. And so it's actually quite challenging to find a suitable promoter for that. Yeah.
Okay. Yeah, no, thank you very much. But yeah, so this is all super beautiful work and very impressive. Thank
Thank
you.
you.
Anybody else have a question? Oh, Manon. Oh, your microphone is off.
Sorry. Thanks a lot for the talk. It's a little bit of a detailed question about the tomogram. The whole conoid is surrounded by a dense compartment with very big black condensates. Do you know what this is?
Actually, no, I don't really know. And actually, I wonder if those are ice crystals. I'm not sure. Yeah, I'm not sure what they are, but they seems to be outside of the cell.
Okay. Okay, I see. Thanks.
Edmée?
I have a very practical question. So I've been doing proteomics on Symbiodinium, and I was struggling because I only had transcriptomes, and then we just translated it. But the problem is that the RNAs are several RNAs together, and then only when it's translated, it's cut into several proteins, and it happens a lot apparently in dinos. And does it also happen in Perkinsus? And
No, it's not the case in Perkinsus. Perkinsus has a more canonical way of making like proteins. It's not as complicated as dinoflagellates.
Okay, all right, thank you.
Yeah.
Interesting talk.
Okay, Lily, go ahead.
Sorry. I was quite impressed to see that you have 6,000 new genes for the genome.
Yeah.
Yeah. How can you? I kind of didn't listen properly at that point. Can you explain how you got to
Yeah,
that
so
briefly?
basically the previous genome was just like from what I understand, because I'm also not an expert in like genomic. So it was just like an in silico genome. And I think there was only support from one RNA-seq dataset, but as this dataset has been done only on trophozoite and Schison stages. So basically there was no RNA support for the hypnospore stage, the sporangium stage, and the zoospore stage. So all the genes that were expressed at those stages had no RNA-seq support, and this is why they were so badly predicted.
Oh, wow.
And now that we have like a very nice RNA-seq on all those different stages, it's way easier for like the program to predict the genes from those RNA data.
That's interesting. Yeah. Yeah, no, that's amazing because the genome was really quite small and yeah, not great. So I'm really looking forward to your genome.
I have a question for you. I was wondering, I know there's kind of a vast diversity of Perkinsids and a lot that we maybe haven't explored yet. But I don't really know much about sort of like the morphological and structural differences across the group. How much would you expect to see these structures conserved or not conserved across the Perkinsids more broadly? Because I mean, I know they infect so many different hosts, and yeah, there's a question coming from major lack of knowledge of this group, but
Yeah, no, that's a good question. Actually, I'm not exactly sure. So there is two groups that I showed in the introduction, the one that infect the fish and the one that infect the tadpoles. Actually, I think there is almost no cell biology done on those. It's only like EM pictures from quite old papers. So we actually don't know all the cell stages. The one for which we know a little bit more are the Parvilucifera, for example, those parasites that infect dinoflagellates. And they seems to have kind of like a similar cell stages, with especially in terms of like zoospore relation and zoospore formation. So actually, I think that the zoospore in Perkinsus and in Parvilucifera must be quite similar, I would expect. Then into the content of those organelles, I'm not exactly sure. I would need to look at if orthologs of those potentially apical complex proteins are also found in other organisms. Yeah.
okay, cool. Thank you. It looks like we have another question in the chat from Leo. I saw on the UMAP a cluster for plastids. Do you or what processes are found? Do you know what processes are found in Perkinsid plastids? And did you happen to see how many membranes are surrounding the plastid under the ET?
Yeah, so this is going to be a study that is going to come out quite soon. It's not mine, it's someone else in the lab working on the plastid in Perkinsus. So what I can say that it seems that in Perkinsus, the genome, there is no plastid genome. Everything is encoded in the nucleus. It seems that we believe that it's only one membrane because there is no evidence for any translocons. And it seems that there is only the IPP pathway that will be encoded in the plastid of Perkinsus.
Okay.
Wow, very, very cool. Thank you. Also, thank you for the amazing talk.
Thank you.
Yes. So if anybody has another question, speak now. Otherwise, I just want to say.
So I have another question.
Oh, yeah. Okay.
If I may have a follow-up question on the plastids. So without an evidence for translocons and with the plastid seemingly having only one membrane, how do we know that it's evolutionarily derived from the plastid that we got from the primary? Yeah, how do we know that it's a plastid and not something else?
Yeah, that's a good question. I'm not an expert in the plastid of Perkinsus, so I don't want to say.
Yeah, don't worry about it. Yeah,
But
it's very, very curious.
yeah, I'm not exactly sure how to answer.
Fair enough. Okay. So with that, I will wrap up the questions. Thank you all for your questions and engagement, and I will hand it off to Lily to take us out.
Okay. I'll do my best. Yeah, no, thank you so much, the two speakers. This was super enjoyable, and I'm glad I stayed late at work today. I just want to say if you want to talk ever in this seminar, you don't have to wait that we contact you and force you into it, please, you can just contact us via the website. There is an email address, and you will also find our Discord channel on the website. I think the website should be now also in the chat. And we're really happy to host you, and it doesn't have to be a finished project. It can also be a project in progress, maybe sometimes even better, because you might be dealing with some issues someone else might be having or can give you advice. The other thing is, you can join also the Discord channel for discussion, or just if you have any questions. And we are planning to host the next seminar probably in January, so there's lots of time to think about if you want to present. And is there anything else I should mention? We're probably switching the times back and forth, but we will see which time zone works best for everyone. But as I mentioned beginning, we are recording this, so for people who cannot join at this time, hopefully they have access. They can look at the recording. So yes, I hope you all enjoyed it, and we're really looking forward to see you next time, or in the meantime on Discord or via the website.
Yeah.
So
the recording, we will try to make some YouTube channel and then share it on the website and through the email list we have so far.
exactly, yeah. And if you know anyone that might be interested joining the email list, please let us know, and we will add them. So it would be great. Okay, have a nice rest of the day wherever you